Hi, microbiology students. Welcome to Unit 4, which is going to be focusing on Chapter 9 and 10. And specifically, we're really getting into more biology-heavy concepts, which is going to be focusing on genetics in this specific unit. So again, with our Unit 4 introduction, let's go through a little bit of this. And so the first thing is the first two lecture PowerPoints you're going to have is Chapter 9, which focuses on genetics, specifically how DNA is replicated and how we create proteins. Chapter 10 is now going to talk about the genetics and take it a little bit further. What is genetic engineering? What is genomics, which is the study of the genome? And how do microbes transfer their genes? And there's three specific processes we're going to focus on in this chapter is transformation, conjugation, transduction, which is all about gene transfer. So there are two labs that you're going to complete with this unit. The first one is DNA replication and protein synthesis. So these are actually tied to concepts within Chapter 9. So when you're going through the PowerPoint for Chapter 9, I would suggest also going through and doing this lab. This lab is actually the basis for your open response, which I'll get to here in a second. The aseptic culture transfer is not directly tied to either one of these chapters. It's more of a microbial technique and a microbiology technique in order to actually transfer bacteria and grow bacteria. Specifically, it's actually showing you how we would transfer bacteria from yogurt to broth and then growing that, which will eventually move to a solid agar slant. So we're kind of going through this process of transferring bacteria. So again, this specific lab is not tied to either one of these chapters, but the DNA replication and protein synthesis lab is. So speaking of that open response, I'm going to have you transcribe and translate a given DNA sequence. So I'm going to give you a DNA sequence. There's a codon table that's going to be given to you, and you are going to have to transcribe DNA and then take that transcription and translate it into a protein. And so that is actually based off of the lab that you guys have for this unit. So let me go through and show you the unit itself, and then I'll go through the lab itself to show you how to do that open response and what you need to know. So again, 2 lecture PowerPoints. There are some helpful videos because we're getting heavy into genetics. Super useful. If there are some things that you're struggling with, check the videos folder. Then we've got the lab instructions for both of these labs. Once again, the DNA replication, which goes with Chapter 9, and the aseptic culture from a broth tube to an agar slant, which is not really tied to either one of these chapters. So let's go ahead and look at the DNA replication and protein synthesis lab real quick. So when you open up the lab, there are 3 different activities. The first 2 are focusing on DNA replication, which is not the focus of your open response. Your open response is actually focusing on protein synthesis. And there are 2 different processes that we go through for protein synthesis, and that's transcription and translation. So if you go down to number 13, this is pretty much what I'm going to give you. Is I'm going to give you a DNA sequence. You are then going to have to transcribe that sequence into mRNA. And once you have that mRNA sequence, you are going to translate it using the codon table. I will give you the codon table. Do not worry about memorizing the amino acids. Don't worry about memorizing the codon table. I will give it to you. So I will give you a DNA sequence, and the same process you go through in number 13 and number 14 in this DNA replication and protein synthesis lab is what you are going to need to do on the exam itself. So make sure that you are comfortable with the process. Again, don't memorize that codon table. I will make sure that you have it for you. And then again, your exam is always at the bottom. If you run into any questions whatsoever, please reach out to me and let me know. I'm here to help, and I look forward to hearing from you.